becoming their own random strands of DNA?
Because they don't have primers to tell the DNA polymerase to come and start making copies. Yeah, everything in your cell is regulated. So for this process, there's dozens of proteins that regulate this process. And we're on the surface. We're just barely on the surface here. So there's lots more going on. So I'm trying not to complicate things too much. Okay.
DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides.
So that's DNA polymerase 1 removing the primer.
Because the two complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer.
So I'm going to pause it right there.
On the
So,
other side, the Okazaki acts as a primer on the other side. Is that
no, the Okazaki is the new DNA that's being, it's just DNA, it's just called an Okazaki fragment just because it's a fragment of DNA unlike the continuous piece that's made on the leading strand. Yeah, and the guy who discovered it wanted to name them. It's just DNA though, that's all it is, it's just little chunks of DNA. And so you can see as the helicase is moving this way, on the leading strand, you know, DNA polymerase can just keep synthesized and following behind helicase. But on the lagging strand, hopefully you saw that the enzyme came in first and bounded this, and then went this way, and then it's now going to come over here and make this one, and then there's going to be a new primer right here, and it's going to bounce this way and go that way.
is there a certain name for that primer, or is just to know it's a primer?
It's just another primer. It's the same type of primer that's put in by primase. It's just complementary to that piece of DNA instead of the other. All right, so, yeah, question?
The Okazaki fragments, are those the green parts?
The green parts are the primers.
Oh.
So the Okazaki fragment, it's just DNA, which is why it's shown in blue. So this is an Okazaki fragment right there. This is an Okazaki fragment right there. The Okazaki fragment is the pieces of DNA in between the primers.
Oh.
Yeah, on the lagging strand. So here's the lagging strand. You can tell it's a lagging strand because it's got multiple primers on it. And so this right here is the Okazaki fragment that's in between those two RNA primers. Like I said, it's just DNA. Just got a really fancy name. All right, I'm going to back this up a little bit so y'all can see that again. Cool. All right.
Of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
Does that top one turn it into a helix? Is it?
Yeah, so after it's been replicated, yeah, it's just going to go back. So those single-stranded binding proteins, because they're moving, they're following behind helicase. So once the new piece of DNA has been made, those single-stranded binding proteins are gone. So that's what's going to allow the DNA to go back to its helical state.
And when does the bottom one start curling again?
After you join, you have to use that ligase enzyme to join these chunks of DNA together. And so after that occurs, then it can go back to being wound again.
So really, it would have already started curling?
So yeah, technically it should have already started going back to the helical stage. Yeah.
Did the two new
That's the
strands
binder.
separate from the original one?
Say that again?
Like, there's the original strand and then you have these two, how do they?
Exactly, so literally what happens is, again, if we have our, where helicase is going, so here's our, imagine this is, well, I guess I'll just try to draw it first. All right, so helicase is coming and it's going this direction, right? And so it's unwinding, and so eventually you can imagine that as this goes all the way through the piece of DNA, it's literally separating the pieces of DNA. And so that's why we call this semi-conservative, is because this is, let's just say that's the 5' and that's the 3' and that's the 3' and that's the 5'. So your original piece of DNA is going to be incorporated in the new DNA. So you're literally physically separating the two strands and then making a copy of one another. One of these is going to be the leading strand and the other one's going to be the lagging strand.
And then they're separate,
And then they're separate, yeah.
and then they start.
And then the same thing happens once it goes back into S phase and it needs to replicate its DNA again. Now those two molecules, they'll separate. And then the same thing will happen. Make a new copy, make a new copy. So this semi-conservative replication, all we're talking about is after this first round of replication. You end up with a double-stranded piece of DNA, one with the original strand of DNA, and then one made up of the new DNA. That's what semi-conservative replication is. You okay? Okay. Did the video